Thank you for the very kind introduction. I guess I feel figuratively honored. I'm going to talk mostly about brain imaging, and you can rest assured I have mostly pictures. So if you like pictures. I want to give a brief promotional announcement. We at Johns Hopkins, myself, Ciprian, Han Liu, and a couple other people, now Martin Lindquist, have this working group called the Statistical Methods and Applications for Research and Technology, or SMART group. So we're really good at acronyms, among other things. At any rate, I bought a kind of vanity website, so we have www.smart-stats.org. So check out that website if you get a chance. I like to call the people that work there, or work in the group, the Smarties, so you can see who the Smarties are. At any rate, we're very excited about some of the things going on in the group. We have our finger in a lot of pots at this point. So I want to talk about brain imaging, and I want to talk about what is at least thought to be a pretty exciting area of neuroscience right now. That's an old idea. It's received sort of a revolution. And let me start with this sort of thought experiment. That's this guy right here. This green guy is my personal rendition of what I think an alien looks like. And so imagine you're an alien, and imagine you had to figure out what, like, something like a Google data center, what it did. And, you know, so in my thought experiment, your alien computers work in different ways than our computers. So it's a hard problem on your behalf. So bear with me with this. If that was the case, you would certainly, and this is analogous to what we're trying to do with the brain right now. If that were the case, you know, you'd have to figure that something like the wiring that A connects to B physically would have to be important. The other thing that would have to be important is that, you know, maybe when this thing lights up over here, this other thing lights up over there. That's got to be important. And maybe when you operate the computer in one way, it functions in a way in terms of its output. And that's the idea of the direction that neuroscience is kind of taking right now, or a main direction that neuroscience is taking right now, is that this idea of looking at functional and structural and other kinds of connections in the brain. And it's an order of magnitude harder to do. So our problem is actually much harder than what the alien has to do. The alien only has to work with Google's data center. You know, a human brain has many more synaptic connections than a data center would have in terms of physical connections, and even when you think about the actual microcircuitry. The other thing is when we measure brain connections, there's all these different ways that we could do it. You know, we can, and unfortunately, there's some sort of Heisenberg principle at work with the study of this sort of stuff, that you can kind of get good spatial resolution, but maybe low field of view. You can get you know, they even gave a name for if you were to enumerate every single connection in the brain, the so-called connectome, a sort of portmanteau of the words connection and genome, I guess. So the connectome is to the brain what the genome is to to the study of genetics. The connectome is this idea of being able for a human to map every single synaptic connection in the brain, but the resolution we have to look at that in terms of human brains is just so you know, far off from being able to do that. we have to resort to all different other ways to get at these problems indirectly. And then it starts to get hard because you start to deal with trade-offs. You can think about things like functional connections and study them with fMRI. You can think about structural connections and study them with things like diffusion tensor imaging. And so I'm going to talk about some of the ways in which we're investigating this, but I wanted to at least for a minute enumerate why this problem is so hard. And it's also true that our brains have connections or things that could be defined as connections that operate in many different contexts. So, for example, we do have physical connections in the form of neurons, fiber bundles, synapses, and these sorts of things. But we also have chemical connections when, you know, the way our brains work is with respect to these ion gradients and chemical gradients. And these sort of chemical connections are also potentially informative, and then functional connections as well. So I think it's a really hard problem, and there's lots of people that are working on it and looking at it in lots of different ways. And what I'm going to present today is some ways in which we're looking at it. So one way in which we're looking at it is we look at diffusion tensor imaging, which is a way for mapping large white matter fiber bundles in the brain. In this case, what I'm displaying here is a summary of these diffusion tensor images. And what you can try to do, at least, people dispute whether or not you're being successful at actually doing this, is to map fiber bundles in the brain. And what we've taken to doing is trying to come up with ways for parameterizing these fiber bundles, fitting sort of what we like to call tubes, though they're not tubes at all. I think of a tube as like, I don't know, like the center of a toilet paper roll, not so much things like this, but we call them tubes nonetheless. And we try to fit tubes through these fiber bundles and use the parameterization of the tubes to perform prediction. And again, here we're not getting anywhere near this idea of these connectome type problems. We're getting gigantic brain connections and trying to figure out ways to do statistics with them. And so because of that, we only apply these things in settings where we would, where there's large non-subtle damage to the brain, like in multiple sclerosis, with tracts that we would think would be meaningful to study. For example, if we're studying, we're trying to relate the parameterization to vision, we'll look at tracts that are related to vision in the occipital lobe. So I wanted to give another example of this kind of problem, and another one that we're working on, and how it relates to sort of the problem of big data sets. We're also looking at the so-called idea of effective connectivity. So in this example, so I just want to mention in this previous example the work was by Jeff Goldsmith, who's a graduate student in the department there. This work was by Haley Headland, now an alumnus of the department. And what we were interested in, or what the collaborators that came to us were interested in, is they took they took subjects with rather severe epilepsy, and they saw off the top of their head. The skull. They saw off the top of their skull, and they put an electrode plate directly on their brain, right on top of their cortex. So obviously you have to be fairly sick to merit these kinds of treatments. All sort of drug therapies have failed and that sort of thing. And the rationale for putting this so-called electrocorticography sheet on the brain is for surgical reasons. But before they do the surgery, actually, usually the patients are sitting around for a long time. They let us do some experimentation. They let us play some sounds and do the recordings. But to give you a sense of the scale of these kinds of data sets, is they're recording 1,000 and sometimes 10,000 measurements per second. Okay, and then they record this for minutes and minutes and minutes. So you can get a sense of the scale of these kinds of data sets. So one thing that we've been interested in, and this I think was a very nice result because it's quite simple, is that a thing that people are quite interested in is taking and treating every electrode as if it were a node and trying to come up with a directed graph that represents, say when you play a tone and there's sort of recorded neuronal activation at that at that neuron, can you detect that there's sort of later neuronal activation at other neurons? And so the most common technique to do this is the so-called Granger causality, named after the economist Granger, who I believe won the Nobel Prize for this work. The technique is quite simple. It looks at sort of how much of the lagged autocorrelation in a time series is explained by looking at the lagged autocorrelations in another time series and vice versa, and then you just draw the arrows depending on some significance test. Well, if you're to do the standard bivariate Granger analyses, where you take every single pair of electrodes and do some sort of thresholding with false discovery rates or something like that, you wind up with a graph that looks something like that, and maybe we're hamming it up a bit by making it look a little bit too much like a rat's nest. Well, Haley's work was to say to this community that creates a lot of graphs that look like this, well, it's entirely possible, for example, that you're drawing a line between A and C, but really, you know, the electrode A tends to fire before electrode B, and B tends to fire before C, and that it's really this sort of mediating pathway through C that connects A and C, and not some direct connection between A and C. So what she did is just extend it, and the idea is that the data sets are so large that you can't do a giant multivariate Granger causal model. You can't do Kalman filtering and these other things that you might look at these other things you might do to look at temporal relationships in time series. So she said, let's do the simplest possible thing. Let's take, if I see a line between A and C, for example, let me look at all other electrodes that follow this potential mediating relationship where A connects to the electrode and B connects to C. Look at all possible electrodes like that and just fit the 3-variable Granger model and see if the line goes away. Sort of some sort of pruning approach. And if you do that, you go from this giant rat's nest to this much more simple graph. And what was really nice about it is the way the graph is organized is it starts at 17 is sort of this purple area, and the directed relationship is sort of posterior to anterior. And if you look at actually how it's occurring, it does bear out the neuroscience in this area, that you're sort of going from auditory hearing areas of the brain to auditory processing areas of the brain. That you're sort of this patient was sort of hearing the sound, and then they were thinking about the sound, they were processing about the sound, and it sort of works in the way that you would hope that you get a fairly localized early effect in the auditory areas, and it spreads sort of outward towards the processing areas. So we were quite happy about that, and it was just a rather simple technique. And then I want to make another point. Here's some functional fMRI data, and this is to date my favorite fMRI study, and I'll tell you why later. But this is an example. This is a typically developing child here, and this is part of a scale, a neurological scale that someone at Hopkins named Martha Denkla developed, and it's called the PANS, or Physiological and Neurological something scale. That's not the exact name. There's some other letters in there. And what they're doing is they're asking this child to walk on their heels. You see, he's kind of moving his hands while he's doing it. He has these sort of overflow movements. And so we conjectured that we could detect that these overflow movements are sort of related to brain connectivity between different hemispheres of the brain, that there's sort of maybe some idea of excess connectivity between the feet and the legs in the motor cortex for these subjects. So we took resting state fMRI data, so you just stick people in the fMRI scanner and you let them just sit there and take scans. And we took correlations between motor areas, and we looked at, and what was very interesting is that this score, if you look between left and right hemispheric correlations and you take the PANS score, there's a fairly clear gradient between lower PANS scores and more what we were describing as interhemispheric lateralization, and there's a fairly clear gradient going on there, and it satisfies all sorts of multiple comparisons, corrections, and stuff like that. And in fact, in this case, we're not studying the whole brain; we're seeding specific voxels. But the reason I want to bring this up is because the reason why I like it is that the neuroscientists came up with a very, very clear hypothesis as to what should be going on vis-a-vis the brain and the associated brain connectivity, and we looked at when it was there. And most of the things I do don't fall out that way. I'll give you a lot more examples of where we're throwing mud at the wall and seeing what sticks. But what I like about this is we actually had a hypothesis, and it panned out. And later on, I'm going to talk about this competition we investigated. And maybe just in the interest of time, I'll skip that stuff and start talking about the cocktail party problem. So, so that other experiment, this PAN-S experiment, is what I think of as sort of an ideal in the field of fMRI. A very clear scientific hypothesis, a very clear connection between the methodology and the tools you would use to evaluate it, and in that case, a very nice result. What we have been working on mostly instead are much more empirical hypotheses, settings where we're kind of asking the data to do double and sometimes triple duty of generating the hypotheses and confirming the hypotheses. And this tends to be the direction that everything seems to be going with these large data set analyses. They tend, seems to be a movement more towards these sort of generalized factor analytic type methods and asking a lot of the data. And I guess the point that I'm trying to make is just because you have a lot of numbers doesn't mean you have a lot of data. And I'm going to, at the end, show you what I believe to be a fairly good example of that. But at any rate, I want to talk, but I still believe there's quite a bit of use to these sorts of empirical techniques. And this is what I'm working on, so I must at some level believe they're useful. And so I'm going to talk a little bit about ways we're looking at it. And we're kind of following along the lines of this so-called cocktail party problem. So the cocktail party problem is a famous problem where you have, let's say, 3 microphones in a room, and you have 3 sources. Scott's talking to Ingo, Jeff's talking to Ciprian, and someone else, maybe this is Hilda, and Marie or Rafa's playing the guitar for Marie over here. And this microphone would probably pick up Rafa's guitar very well, but maybe would only pick up Scott's conversation very faintly, and so on. And the blind source separator, the cocktail party problem, the goal of the cocktail party problem is to take the recorded mixed signals from the microphones and deconvolve them to obtain the original source signals. And the method in which we've adopted to look at this is kind of not a solved problem, but at least there's, under certain assumptions, you can solve this problem. If you assume linear mixing and some other assumptions, you can solve this problem, and we tend to attack it with these sort of generalized factor analytic tools called independent component analysis. So independent component analysis is a member of a collection of factor analysis tools where you're assuming independence of the underlying factors that you're trying to discover, and you estimate their densities, the underlying densities of those factors, more non-parametrically than you would, say, with standard factor analysis with Gaussian assumptions. So since the name of the talk is large-scale inference, I wanted to at least mention the inferential tool that we're promoting in this area. And I should say, for those of you that don't work in fMRI, this stuff is a juggernaut in fMRI right now. It is everywhere. And the specific instance that is probably most common is so-called Group ICA. A Group ICA, what you do is you take a subject's fMRI image. So an fMRI image has time. You're recording a bunch of 3-dimensional images over time. It has time and it has space. And let's assume that you can restructure the data into a matrix so that you have time on the columns, you have space, voxels or space on the columns and time on the rows. Now you have to be a little bit careful in what you do that, but for the purpose of the talk, let's assume you can do that very easily. What Group ICA does is it just takes all your subjects and stacks them up like this, just stacks them up into one giant matrix. So if every subject was recorded for, say, 500 time units and 70,000 voxels, and you have 10 subjects, then now you have a 5,000 by number of voxels matrix that you're then just sticking into a factor analytic model. And ICA is the specific factor analytic model that takes this collection of green bricks. I do most of my math via bricks these days, and decomposes it into a collection of lower dimensional orange bricks times a blue matrix here. So think, you know, these green things are on the order of, say, 200 by 100,000. The orange ones are, say, on the order of 200 by 30. This blue one is on the order of 30 by 200,000 or something like that, okay? The way that the neuroscientists have a tendency to interpret ICA is, Group ICA, is that, well, this blue matrix is, every row of this is a spatial map. Every row of this is a spatial map. And so they tend, and they're common across subjects. So every subject gets their own orange brick, but everyone has a common blue brick. So the way that the neuroscientists like to interpret this is they like to say that, well, the blue bricks sort of, because ICA is in essence kind of working on is similar to singular value decompositions, sort of rooted in covariance decompositions and so on. The thinking of these blue rows as what they would call brain networks, and they think of the columns of these orange bricks as sort of how those brain networks are sort of weighted over time to yield the observed data. That's how they think about it. Now, what that requires you to do is to associate an output from a mathematical model to a scientific interpretation of a so-called brain network, an entity which is not entirely well defined to begin with. So I tend to not think of it this way. I tend to think of it more as it's a parsimonious model that assumes some parsimony by assuming not everyone has their own blue brick. They just all have a common blue brick across subjects, and that is a parsimony assumption that yields a factor analytic model. But let me go through some examples of that. Well, I guess before I go through some examples, let me tell you what this is very common in the fMRI literature. So what I'm covering right now is stuff that's extremely commonly done. Our contribution in this area is to talk about, well, you know, don't just do Group ICA, the factor analytic model. Let's talk about how you actually do inference based on the output from Group ICA. And we basically say, take these time courses, because you know, this time course from this person and this time course from this person correspond to the first spatial map that is common across people. We're assuming that at some level, this time course and this time course and this time course are interpretable across subjects. So we say, grab those time courses, which are functions over time, and model them using functional logistic regression models, sort of using kind of case control thinking. So we even in most cases, we might have specifically the number of members of each group fixed by the study design, but we'll use some sort of logistic regression model and relate group status to these ICA decompositions via a functional regression model. And to do, typically when you do these functional regression models, what you then do is some sort of principal component decomposition, some sort of decomposition of the functional regressor. And so that's what we do. So we do, in essence, what we're basically saying to do is take your fMRI time series, stack them up, do Group ICA, take the time weight matrices, stack them up, do a PCA, take the PCA scores, and stick them into logistic regression. If I were to summarize our entire method, well, that's not even a summary. That's exactly what our method is. But you know, we can mathify it a bit. At any rate, so here's an example where we think we're getting reasonable things when we apply. This by the way, I think I could argue that this is useful for paradigm-related fMRI, but not so much so for resting state. But here's an example where we took 13 age and gender matched subjects, and 13 of them had mild cognitive impairment as governed by some form of cognitive function testing, and 13 of them were controls. And we threw 26 subjects into this Group ICA model, took the weight matrices, did a principal component analysis of the temporal weight matrices, and then took the principal component scores and put them into a logistic regression model. And because we only had 26 subjects, we did an exact form of logistic regression on it. And these are which results in a permutation test. These are the permutation test p-values. So we get some ones that kind of consistently come out. Basically, the 11th, 13th, 19th, 22nd, 26th, and 28th spatial maps. And so here's kind of, if you were a neuroscientist, where those spatial maps tend to localize in the brain. And there's some interesting things going on. And again, this is one of these cases where we're using the data to both discover the so-called brain networks and then evaluate their importance at the same time. So you have to take all this stuff with a grain of salt. But then we do take these spatial maps and map them back to what we read in the literature. And we see that, you know, say, for example, ICA 11 kind of loads on things like the temporal poles and cerebellar areas, but more importantly, the temporal poles. And then we go into the Alzheimer's disease research and look and see to what extent the temporal poles are indicated in paradigm-related fMRI as an early stage biomarker for disease. And we see a lot of these things are very common. We see that the temporal poles come up a lot. The funnest one that seems to localize the most clearly is these olfactory areas. And it seems like smell memory is actually a very common thing that you lose early on in Alzheimer's disease. So much so that the investigator that we were working with in this had tried to design a smell fMRI paradigm, but they couldn't get, the olfactometer, as it's called, to actually work. So this is the example of the kind of output that you get. These are the weight matrices across all subjects for this spatial map. This is the spatial map that you get. These are the weight matrices. So it's basically a principal component decomposition of this matrix that we're then putting into these functional regression models. And again, you have to be concerned with what we're doing because we're using on the order of a couple 100,000 measurements to predict 26 zeros and ones. And we're cognizant of that fact. Here's another study. So just to give you another example of how we're doing this with again, some undirected hypotheses, but how at least there's some hope that there's something interesting. A friend came to me with some former Iraq war veterans and had 9 of them who had had some sort of traumatic brain injury and 9 controls and did resting state fMRI. And in this case, resting state fMRI or resting state EEG or resting state whatever is all that you can do. You can't ask these people with a traumatic brain injury to engage in a paradigm. So what's interesting about this is it does work out that if you throw these group of patients into Group ICA, you get these brain networks coming out like the default mode network, the network labeled the default mode network, that come out very commonly across lots of studies. And before he even contacted me, this investigator had said that maybe it's the default mode network that seems to be indicated in what predicts when a person has a traumatic brain injury, whether or not they recover. Now, again, I think if you press him hard enough, whether or not the scientific underpinnings of why looking at the default mode network in that case don't seem to be there, and the default mode network is the thing that everyone's looking at for every disease. So it's not at all clear as to exactly how tight this hypothesis is. But nonetheless, if I then were to say take these various brain networks and do the kind of testing that we were doing before with them, we see some things that when we compare persons with traumatic brain injury and controls. So I wanted to put a plug now for something that we just finished. What we'd like to do now is we believe enough in these Group ICA methods to pursue them on a larger scale. And we think now that people are starting to do fMRI, resting state fMRI in very large-scale studies with thousands of subjects or at least hundreds of subjects, that you need tools to be able to run ICA on large numbers of subjects with, kind of modest computing infrastructures. And so Ani, she has an algorithm which we've taken to calling HDICA, where we basically say, not unlike you can do with the singular value decomposition, you can do with ICA. You can execute ICA in a way that you never have to actually load this full concatenated data matrix into memory. You can do calculation using only loading a brick at a time, basically is what it boils down to. And with that, then at least in principle, the algorithm is arbitrarily scalable, as long as you're willing to let your computer run for a long time. So as an example, this is running and she's actually using her specific ICA algorithm in this, but the principles of what she's doing would extend to other ICA algorithms as well. She's been able to run ICA right now on 150 subjects, which as far as we know is the largest run of Group ICA that anyone's ever done. But maybe if someone proves us wrong, we'll run it on a bigger subset so we can make that claim in the future. But anyway, we think these kinds of ideas are sort of more of what's necessary to push this area forward into the kinds of data sets that are being generated now. And I just want to mention that her ICA algorithm actually outperforms what is the classical ICA algorithm that everyone uses. How am I doing for time? About 5 minutes. Okay, in my last 5 minutes, I want to talk about this competition that we engaged in. And this is a kind of a cautionary tale. There was this ADHD 200 competition where they people at NYU and WashU and then some very kind folks at the Neuro Bureau put out some processed data set where they gave out 776 subjects. And the data set was a bit of a mess because many subjects had multiple scans. But they had resting state and structural fMRI for 776 subjects. And then they gave out 197 people where they gave out imaging data, but they didn't give out the majority of the covariate information, and specifically they didn't give out diagnosis. So in this case we have 776 people, 491 were typically developing children, 163 were diagnosed as ADHD of a specific kind, 11 were diagnosed as the hyperactive kind, and 111 were diagnosed as the other kind. I have the notations on the next page. And then for the 197 of them, they gave us no diagnosis. And the goal was to use the 776 to train your algorithm to then predict the remaining 200. Send in your predictions. They have some rule for determining who wins. And the goal was to be able to use this, in this case, 19 billion numbers to predict 200 zeros and ones. So there's a slight potential for overfitting. And we had other covariates too. So we had, and the covariates were hugely important. So we had majority male in the study, largely because this was ADHD studies, and ADHD tends to be more prevalent in males. So presumably males were oversampled in the sites, at the sites. Ages, between 18 and 20 mostly, with a few other older kids. Most of the people were right-handed, of course. And this IQ measurement puts most people in normal-ish scales, but at least with some variation in IQ. This is an IQ summary. We also had, for different studies with different amounts of missingness, different kinds of IQ measurements. What was probably the most interesting variable I found was the site that the data came from itself. So for example, Peking had 47% controls, 12% ADHD combined type, 0 hyperimpulsive, and 20% inattentive, and then the 21% withheld where you had to generate the predictions. But this is hugely informative data. For example, if you look at PIT, 100% of the data where you have the actual classification were controls. And you had to predict these other 9% of the PIT data. you'd be silly, regardless of your information, to put those people as anything other than controls, especially given the competition rules, which penalized you heavily for declaring someone ADHD that was not. And then they were smart enough to, for the Brown data, to not give you anyone except you have to predict there. So you have to, if you come up with any way to use site, you have to figure out a way to then not use site when you're predicting for the Brown data. So at any rate, I'll tell you the long story short. For us, the best imaging predictor was if you were to cluster the motor network into 5 areas. This is our 5 clusters, and simply take the correlation in the time courses between those 5 clusters. That yields 10 numbers. Take those 10 numbers, put them in a machine learning algorithm. That was the best imaging predictor that I came up with. Ani came up with what turned out to be the best imaging predictor overall. Was she, for every subject's fMRI data, did the CUR decomposition, took these CUR decomposition probability maps, concatenated them across subjects, and came up with a rule for picking which voxels survived the CUR decomposition aggregation, then took these associated voxels, took their correlations, and dumped those correlations into a machine learning algorithm. That was those are the 2 things that we know the most. So the machine learning algorithms we used random forest. Here's random forest. It's obviously not the math part. This is boosting, and this is actually, someone drew a cartoon of support vector machines. This is a kid creating a separating hyperplane between his parents who are arguing. So here's from our report. I compiled the data with 4 predictions. One was random forest, basic demographic data, and the 10 correlations. Han, the computer scientist / statistician in our group, plus his 2 students, used support vector machines plus basic demographic data and those 10 correlations. Ani used the CUR decomposition, and John Micheli used random forest boosting and support vector machines all together with the basic covariate data. And then John did something really funny. He was our person for looking at the motion data. We conjectured that ADHD kids would fidget in the scanner a little bit more. So we figured out ways to get motion data and put the motion data in, figuring that it would be good predictors. But they were on to us, the competition organizers, because the test set that they selected was selected for having very low motion. So the motion, so unfortunately John's, which we went into thinking it would be the best predictor, actually was the worst. Internally, it was the worst. We actually can't evaluate anything externally, but I'll give you some external numbers. So internally, the algorithm 1 had 75% accuracy, or basically accuracy. This is relative to competition points, but let's just think of it as accuracy. Ani's did the best with 78% internally, and John's was at 72%. If you do a very brute force and ignorance thing where you just declare everyone a control, you get it about 65%. So we're not adding all together that much. If you do brute force and ignorance, you get 65%. If you ignore 100% of the imaging data, you do about 72%. So we're adding 19 million numbers, several months of processing. You know, I think I had every computer except for maybe those, every processor except maybe those in my printer running this data at some point to get an extra couple of percentage points, which we later found out might have been overfitting. So at any rate, we won the competition nominally, but I'll tell you in a minute why I say nominally that we won. So overall, there was a kind of fairly modest improvement over all prediction entrants over just chance, with the best. This is all from the competition organizing site because they haven't released the test data for us to reproduce these results. But the site that seemed to produce the best predictions in terms of being better than chance was the OHSU, which is Oregon. These are the kinds of mistakes that people made, and the big mistake you didn't want to make in this competition was classifying typically developing controls as diseased. And that's actually how we won, is we basically started, we said, let's take the algorithm where we declare everyone a control, and we have to beat that before we before we put anything in. So I was fully committed to giving them a vector of zeros if we couldn't beat it. And so we barely beat out a vector of zeros, but among those that we classified as ADHD, we got their subtype correct quite frequently. Okay, let's see. And then, but here's the moral of the story. There's this team from University of Alberta. So that's how we got declared the winners, because we had very high specificity, and among those we declared as diseased, we got the vast majority of them, 94% correct diagnoses, subdiagnoses, given that we diagnosed people. There's a team from the University of Alberta that used no imaging data whatsoever, and they got disqualified for it, that actually had a higher number of competition points for us. So actually, here's the Alberta team at 121 or something like that, and we're at like 116. So that's for total competition points. And then you see, they had a slightly higher prediction accuracy. We had the highest specificity. We had very low sensitivity. We're almost at the bottom at sensitivity. But then this sort of conditional accuracy, given that we diagnosed people, ours was quite good. Anyway, it was a lot of fun, by the way. So anyway, everything's a bit of a mess. So this goes back to my point that the point of this competition was throw 19 million numbers at 200 zeros and ones and see if you can improve on classification. But my point is, I think we maybe should work a little bit more with the neuroscientist about tightening the hypotheses to see if we can do a little bit better. And then I just want to acknowledge, of course, Dr. Efron and the conference organizers, and especially Hilda, and then all of our prediction group people and all the people that work, all the people that work with the various labs that we work with, and then the competition organizers. These guys, I've talked to them. They're just fantastic people. They are really, they are really heavy promoters of statisticians. They've done the 1,000 Connectome project, where they've put over 1,000 fMRI scans online, freely downloadable. And a big part of why they did that was to enable statisticians where this area has a very high barrier to entry. And they cited that specifically as to why they did that. They put on this prediction competition primarily, among other things, to engage statisticians and computer scientists to get them involved in this area. So I think these people, though many people in the statistical community may not know them, they're tremendous friends of our community. And then of course, NIH, specifically NIBIB. And then I'll just say, do anyone have any questions?